Okay, let's talk evolution of early echinoderms to the later echinoderms. Why not? Yeah. So if we're looking at things that are around pretty early on, like present in the Cambrian, these are all present at around the same time, but remember we have that gap from like 541 till 514, 515, where we don't have great intact body fossils of much stuff. And so echinoderms were undoubtedly doing things at this time. And you do find little disembodied plates just sort of scattered around that could be from echinoderms or something else. Um, but it is, yeah, it's hard to say exactly what they were doing. So while this tree is hypothesizing that one of these things is kind of like an earlier ancestor to the others, or that they break off from the family tree at different points. Really, these things are mostly found around the same time. And so this sort of branching has to have happened a bit before, in that 20 to 30 million year window, before we find the first fossils of these. Yeah, so there's your grain of salt to take. But here's the idea: when you build family trees out of these using just their features and like what's the simplest way to explain how these features are related, what you end up with is always at the bottom you have these tenocystoids like this. And so those always show up as the earliest branch. They have the fewest of these derived features. The next branch, you're going to have things like cincta. Another early branch is going to be, and coming next to cincta usually are going to be your different stylophorans, maybe coming off of cincta later. I think most of the stylophorans are Ordovician, and these are Cambrian things we're looking at here. You'll get some solutes, and they're at least similar to these others. Then they've got your, so the idea is that you start with bilateral symmetry and then you move to asymmetric. And then maybe they move to triradial, like threefold symmetry, kind of, but twisted around. And then they move to fivefold ish symmetry, which does tend to be what you get on some of these other Cambrian things. Some of these stromatocistides, difficult to place because it's a bit like an Asteroid, a bit like an Echinoid, probably not either of those things. And so there's a lot of problematic taxa down in the early Echinodermata. Here's another one of those phylogenies, and it's just showing that, yep, tenocystoids are the earliest thing. And what I like about this one is that it's showing when certain features develop. So the idea that pharyngeal gill slits are found out in tunicates and craniates and cephalochordates, and xenoturbellids and hemichordates. Um, uh, so those may have been ancestral. And so when we get those grooves on a cornute, those may actually be related ancestrally, phylogenetically, to the gill slits that you have on an early baby human or on a modern fish, yeah, for that matter. And then as you go along, you've got points where these different features look like they're developing. So we've got evidence for a water vascular system in development for everything past this. So that seems to be one of the markers, but you can see that's actually before we branch off the hemichordates. So this reconstruction would say that, yeah, maybe that's developed actually before the hemichordates separate from the true echinoderms. Should we even call all of these things echinoderms? Or are these all in a crown group and they're not really echinoderms until we're at like this branch, like with the crinoids? You could make that case, and I don't think it would be a popular case to make right now. It's not the current understanding, but you wouldn't be, it would be hard to say you're absolutely wrong. Yeah. And scientists love saying that. I don't know if we love it, but it's nice when you can actually have the data that actually backs up a statement as strong as that. Here's only looking at things and where they appear in the Cambrian. So these are the different series and stages of the fairly early to mid, like early mid Cambrian. And so you can see all these different groups, and they're showing you with the little letters. This is a little picture of them. And so you can see we've got a lot of these groups that are around at about the same time. Helicoplacus is very early for these, but we don't have great fossils of echinoderms from before that. So is it the fact that they didn't exist, or is it the fact that they didn't fossilize, or is it the fact that we just haven't found the fossils yet? I kind of think that's probably true, but hard to say. And it may be that the conditions at that time did not lend themselves to fossilizing echinoderms. Because if you have a slightly more acidic ocean, that would dissolve something like sterium really, really, really fast. And if the oceans are acidic for several million years, then it may be really hard to get a fossil of something like an echinoderm. Just because it's only the bone parts that fossilize, and the bone parts are very easy to dissolve. In terms of development, this is kind of interesting. All these echinoderms that we know of today and have like solid evidence for start out very bilaterally symmetric. They're proper bilaterians that just lose all semblance of bilateral symmetry as they grow, or they'll retain some elements of it, but not very carefully. And so these are going to have, if you're looking inside the embryo, along the axis, you'll have an axocele. So like a little pouch that's internal. And there's a right one and a left one. And the right one atrophies and more or less disappears as the creature grows. And the right one is going to expand, and it's going to essentially become the digestive tract and stomach. So that's neat. And likewise, you're going to have these two little blue bits. So that's the hydrocele. This is the part that's going to develop into the water vascular system. They start out as two separate ones. One of those is going to atrophy and just go away completely. And so that is going to be the right one is going to disappear. And the left one is going to expand and essentially surround the gut and form the water vascular system that's going to then make tube feet inflate and deflate in all the ambulacra. And so you could look at echinoderms as being extremely, almost pathologically left-handed if you wanted. Because the right hand is just atrophied, like not a hand, just an embryonic part. But yeah, very left-sided. And the left somatocele is going to end up being massive. The right gets really shrunken down. And then these are going to develop into some of the other organs and internal parts. Yeah. And so if you're looking in terms of where do these things end up, so the hydrocele is going to end up running along the top of all these. So this is like a reconstruction of a camptostroma, and reconstructions of some of these other weird things, reconstructions of little echinoids and things like that, early versions of these. But the idea is that, yeah, it looks as though they're all following some version of the same developmental pattern that we see in all the modern groups of echinoderms. So this extreme left-handedness is probably something that goes all the way back. And it's just like, I wouldn't call it a choice that was made, because these things are not deciding whether their genes, survive, break, come to dominate or whatever. But this left side hyperdevelopment and right side atrophy in the embryo appears to be a very, very early echinoderm trait that leads to much of the weirdness that happens afterward. After that point, after you murder one side of your embryo, essentially, or minimize its contribution, any other symmetry is just a front, because you're asymmetric at the core. Yeah, and you know it. All right, so whew, great job. Now let's practice. And I think this is something we'll do in the class. I'll just show them up here. And so Dora the Explorer style, you can pop up and say, it's an echinoid or whatever, whenever you want. Just like point to the screen, get your grubby little fingers all over your computer screen, please. And we will repeat this in the class. So I'm not going to give you the answers, although they're kind of written in the captions below, but they're going to be too far away for you to see in the class. So whatever, study up, practice up. Yeah. So these are echinoids. I'll give you that one. That's for free. Look at these. Oh my gosh. These actually say the name underneath them, which I probably need to cover up and which I'm going to cover up when we get to class. But yeah, you can see them. They look. Some of these classes don't really exist anymore. Like we would call this starts with a C, and it's not a tenocystoid. I can't remember the name. I'm not going to tell you the name. Yeah, it is in the class Stylophora, but remember that's broken into two groups. So is this a mitrate or a cornute? I don't know. Boy, I wish I could tell you. Oh gosh, this one looks kind of like a crinoid, but with kind of like a fat, short stem. Yeah, and not really very crinoid-like at all. Just kind of superficially crinoid-like. Something like, yeah, you might find at the dawn of crinoids, just like the name of it says. Yeah. Here's some other things. So for this, you got to imagine there were some plates that would fill this in, just like that. And you got, ooh, that's pretty buff looking tail like thing right there. I think that could be a stylophore. Now this one doesn't quite look like a stylophore to me. It's not articulated the same way a stylophore would be. And so this one's not a stylophore. But who has a tail that's not a stylophore? I don't know. Great question. Who's this with a little rim of plates around? So that could be a couple things. But with the five-pointed star of ambulacral grooves in the center and five little mouth plates that can open up there adorably. Say ah. Yeah, all right. Good job. Good job, Asteroid. Yeah. And then you go into some of these crazy things. Is it a cystoid? That's a word that we don't really use much anymore. Is it a rhombiferan? That's another word that I dislike. Is it a paracrinoid? Probably not yet. Is it in the stem group that leads to all these things? Maybe. Is it a diploporite? That's another word that doesn't really have a good, firm biological meaning. It's just things that look like this but are not that close related to each other. So, yeah, it's like calling a cloud that looks like an animal an animal. It's not. It's still just. It's mostly a cloud. More cloud than animal. Yeah. Ooh, who's got twisted little plates? And if we could look at these, look, there's one, two, three little ambulacral grooves meeting up at a central mouth. I think I know who that is. Who's this with a little comb on top? Who's this with a little belt of plates that's not a stylophore on the bottom, but a little belt of plates around the outside? Who's this living free and easy out there? Yeah. I don't know. Yeah, and this one, it's almost got no stem, but it's just like a little cup with awesome long ambulacral arms. So, yeah, see if you can figure it out. And there's some more. Echinoderms are great. The echinoderms are weird. Echinoderms are, they just look very strange. And I love them as fossils, and I have a whole bunch of their fossils. So we're going to hit this one pretty hard in the class. So enjoy that. And then the last couple of slides are just there for you for studying or for little questions that we may ask. I did my best to make a little chart. If you can find something that's wrong with the chart, I'd be happy to fix it. It's just me eyeballing these things and trying to get it all like, yeah, roughly in shape. And this is not all of the things that we covered, but it's some of them with some of their features, if that's easier than a chart. I did keep the color codes, I think, consistent throughout. So Asteroids are always a nice sort of forest green color. Yeah. So enjoy that. And that's it. That's it for generalized echinoderms. We'll have other stuff on crinoids because crinoids are also awesome because echinoderms are awesome. That's how the song goes. All right. Thanks very much.